Imagine you are typing out a text message, like a really important one. But right as you hit send, you realize there is a terrible, highly embarrassing typo right in the middle of it.
Oh, the absolute worst feeling.
Right. And obviously, you don't want to throw away your entire phone just to fix that one word. You're not going to replace the whole operating system.
No, definitely not.
You just want like a tiny invisible cursor to go in, find that one specific letter, backspace it, and type the correct one before the message is actually read on the other end. Well, today our mission is to explore a breakthrough that does exactly this, but, inside your body.
Yeah, which is just mind-blowing.
It really is. We are diving into a dense, incredibly detailed 2025 paper from Nature Communications. It's by Wenjian Han and a team of colleagues, and they've essentially built a microscopic text editor for human genetics, and they call it ProAPOBECs. So we're going to unpack the mechanics of this and translate some, frankly, incredibly heavy biochemistry into a story that could literally rewrite the future of medicine.
It really is a remarkable piece of science.
Yeah.
I think since our listeners already follow the biotech space, we probably don't need to labor over the basics too much, but it definitely helps to anchor this and the fundamental difference between DNA editing and RNA editing.
Right, yeah, the classic hard drive versus RAM analogy.
Exactly.
So for you listening, your DNA is the permanent hard drive. It holds the master blueprint for everything in your body. If you use traditional gene editing, like CRISPR, to edit the DNA, you are basically taking a molecular buzzsaw to the permanent hard drive.
Yeah, which is terrifying if you think about it.
Right, because if you make a mistake or if the tool accidentally edits the wrong file, which in science we call off-target mutations, that mistake is permanently coded into your system.
Exactly. And RNA, on the other hand, is like the computer's RAM. Or maybe think of it as a temporary printed copy of a specific blueprint.
Just a temporary printout.
Right. Your body is constantly printing out these short-lived RNA messages to tell your cells what proteins to build. So if we edit the RNA instead of the DNA, we aren't changing the permanent hard drive. We are just applying like a temporary software update.
A patch, almost.
Yeah, a patch. It's transient. It's much safer because if we stop the treatment, the body just goes back to its original code. No permanent buzzsaw required.
Okay, let's unpack this. The scientists call their base editing system ReWire, which stands for, let me check my notes, RNA editing with individual RNA binding enzyme. And it really relies on a two-part harmony. You need a targeting system to find the typo and an editing system to fix it. So let's talk about how it actually navigates the chaotic soup inside a human cell to find one single letter.
So the targeting system relies on something called PUF proteins. Now, PUF proteins are naturally occurring, but the researchers engineer them to act like a highly programmable molecular zip code.
A zip code, I like that.
Yeah, and physically, a PUF protein has this long curved structure made of 8 to 10 repeating motifs. You can think of each motif like a single highly trained bloodhound. Each motif is specifically designed to recognize and bind to one exact genetic letter on the RNA strand.
And what's fascinating is how it actually binds to that letter, because it uses something called the Watson-Crick edge.
Yes, the Watson-Crick edge.
For you listening, picture the RNA letter not just as a flat shape on a page, but as a three-dimensional chemical face. It has a specific pattern of hydrogen bonds sort of poking out of it.
Yeah, it's very textured.
Right. So the PUF protein essentially runs its hands over that face, reading the hydrogen bonds to ensure an absolutely perfect lock and key match. So you program the PUF protein to find a specific 10-letter sequence, and it will just scan until it finds exactly that address.
And that gives us our pointer.
Oh.
But once you find the address, you need the pen, or, well, rather the eraser.
The thing that actually makes the fix.
Yeah, exactly. For the editing side of the equation, they use a family of enzymes called APOBEC. Now, in nature, APOBEC enzymes are fascinating. Their day job is actually immune defense.
Wait, really? Immune defense?
Yeah. They naturally fight off retroviruses, like HIV. When a virus invades a cell, APOBEC enzymes swarm the viral genome and rapidly mutate its genetic letters.
Yes.
Specifically, they change the letter cytosine, or C, into uracil, or U.
Okay, C to U.
Right. They basically scramble the virus's code with so many mutations that the virus is destroyed.
Wait, let me stop you right there, because this is where the logic seems, well, frankly, dangerous. You're telling me we are taking a molecular machine whose entire evolutionary purpose is to carpet bomb viral genomes with massive, chaotic mutations, and we are just setting it loose inside human cells.
Yeah, it sounds like a terrible idea on paper.
How does that not just melt our own RNA and cause widespread cellular panic?
That is the exact right question to ask. And what's fascinating here is that this was the primary roadblock for the entire field of RNA editing. Native APOBECs, like human APOBEC 3A, are overly enthusiastic.
Overly enthusiastic. That's a nice way of putting it.
Very much so. When researchers first tried using them, they caused what we call bystander edits. The PUF protein would drag the APOBEC to the correct target, but once it got there, the APOBEC would just start changing every single C in the vicinity to a U.
Oh, wow. So it's not a sniper rifle at all?
No, it was a shotgun blast.
Yeah.
And to make things worse, natural APOBECs have a very rigid work requirement. They usually only operate in what is called a UC context.
UC context, meaning what exactly?
It means they will only edit a C if it happens to be sitting directly next to a U on the RNA strand. If the C they need to fix is sitting next to an A or a G, the natural enzyme is chemically incapable of making the edit. It just, shrugs and walks away.
Okay, so let's do a quick recap here, just a bullet summary of where we are before we look at how they fixed this mess. Number 1, DNA is hardware, RNA is software. Number 2, the system uses PUF proteins as a programmable address finder, the zip code. Number 3, it uses APOBEC as the CDU letter changer, the eraser. You can use the mnemonic, PUF points, APOBEC alters.
I like that. PUF points, APOBEC alters.
But this first generation tool was messy. It was causing massive collateral damage with bystander edits, and it flat out refused to work unless the typo was sitting right next to a U.
Which makes me want to ask you, the listener, if you had a microscopic editor floating inside you right now, perfectly safe, what genetic typo would you want it to find? Because we clearly needed a better tool to make that a reality. So how did they actually stop the enzyme from shredding the rest of the RNA?
Well, they had to physically rebuild the machinery from the ground up to make it safer. They started by looking at the zip code, the PUF protein. They analyzed its three-dimensional molecular structure and noticed that the original PUF domain they were using was quite rigid. It was basically missing a tiny but crucial structural element called an LP peptide.
Right, and just for context, LP stands for leucine and proline, which are specific amino acids.
Yes, and the key thing about that specific sequence, that leucine and proline, is that it acts like a flexible hinge in a protein.
Oh, a hinge. Exactly. So the scientists thought, what if we insert this LP hinge back into the structure? They did, creating what they called EPUF10, or the enhanced version. It gave the targeting system just enough flexibility to grab the RNA more securely.
And it worked.
It worked beautifully. When they tested it on a glowing green protein called EGFP, the editing efficiency jumped from 69.7% to 82.3%.
So they essentially added a molecular shock absorber to the targeting system, and it immediately worked better. But that still leaves the shotgun blast problem. How do you tame the APOBEC eraser so it only makes one specific edit?
To do that, they really had to understand why it was gripping the RNA so aggressively. It turns out APOBEC 3A enzymes naturally like to form dimers.
Dimers, like pairs.
Yeah, that means two of these enzymes find each other and link up to form a pair. When they pair up like that, their combined electrostatic grip on the RNA becomes incredibly strong, which is exactly what leads to those messy bystander off-target edits. They stay attached way too long and just keep editing.
Okay, so the goal is to stop them from holding hands.
Exactly.
Yeah.
And the scientists did this with some incredibly precise surgical strikes on the protein structure. They targeted specific amino acid sites. For example, there are these spots called H11 and C1781.
H11 and C1781.
Right. And these basically act as the sticky patches or the glue that holds the two enzymes together.
Yeah.
By mutating those specific spots, they neutralize the glue. The enzymes couldn't pair up anymore.
That's incredibly clever.
And they didn't even stop there. They also mutated two other spots, K30 and H56. These are the physical points where the enzyme grabs onto the RNA strand itself. By altering these, they effectively weaken the enzyme's grip just a little bit.
Wow, so you stop them from clumping together and you make them slightly more slippery. That way they get in, make the one edit, and fall off before they can cause collateral damage.
Precisely. They called this upgraded system CU Rewire 4.1. And the data backing this up was undeniable. They ran whole transcriptome sequencing, which means they literally look at every single piece of RNA across the entire cell.
The whole thing.
The whole thing. And CU Rewire 4.1 maintained a very high rate of precision on the exact target they wanted, but drastically reduced the rogue off-target events everywhere else. They successfully turned a molecular shotgun into a sniper rifle.
Which is an incredible feat of biochemistry, but and this is a massive but, they were still stuck in that UC context trap, right?
Yes, the dreaded UC contest.
Even with this perfectly precise sniper rifle, the physics of the enzyme meant it could still only fix a C if it was sitting next to a U. If a patient has a severe, life-threatening genetic disease that is caused by a C sitting next to a G, this beautiful, highly precise tool is completely useless.
It is the ultimate frustration in biology. You perfect the tool, but the tool is still limited by the rigid rules of nature. Which raises the question, when nature's tools aren't good enough for modern medicine, how do we invent entirely new ones?
And this is exactly where this paper blew my mind. Because the answer is artificial intelligence. Specifically, AlphaFold 2. I mean, we've all heard a lot about AI chatbots, but AlphaFold 2 is a system that predicts the highly complex three-dimensional folding structures of proteins based solely on their amino acid sequence. To invent an enzyme that literally does not exist in nature, you have to simulate how it will physically fold.
Yeah, and to understand how they use AlphaFold 2, we really have to look at the architecture of the APOBEC enzyme itself. It's built a bit like a sandwich.
Okay, a sandwich.
Yeah. In the very middle, you have a highly conserved core. This is the actual deaminase engine, the part that performs the chemical reaction of turning a C into a U. And flanking that engine, on either end, you have the N-terminal and C-terminal domains. These outer domains don't do the editing, they act as a structural scaffolding that holds the engine in place.
Great. The researchers used a great analogy here. It's like a molecular Mr. Potato Head. They decided to keep the main body of the scaffolding from the human APOBEC3A enzyme because they knew that scaffolding played nicely with human cells. But they used AlphaFold 2 to predict what would happen if you popped out the middle engine and seamlessly swapped in the core engine from a completely different animal species.
And you can't just mash these proteins together in a lab and hope they work. If the chemical bonds don't align perfectly, the protein just falls apart.
It's useless.
Exactly. That's why the AI was crucial. AlphaFold 2 allowed them to digitally simulate thousands of combinations. They tested core domains from mice, from rats, and from different human variants. They built these predictable, novel enzymes and dubbed them professional APOBECs, or ProAPOBECs.
If we connect this to the bigger picture, this completely shattered the natural limitations. Because by swapping these core engines, they finally broke the UC context rule.
They did.
Suddenly, they had variants that could edit anything. For instance, the AI design variant they called CU5.1 specifically edits a C's next to a G. The variant CU5.15, which uses a mouse core, edits a C next to another C. And incredibly, they found variants like CU5.3 and CU5.17 that are completely omnivorous. They can edit a C sitting next to literally any other genetic letter.
They literally engineered their way out of nature's corner. And to prove it wasn't just a parlor trick, they aimed these new ProAPOBECs at some of the most devastating human diseases in a lab setting.
Like Alzheimer's.
Yes, let's look at Alzheimer's disease. The APOE4 gene is a massive genetic risk factor for Alzheimer's. But what's incredible is that if you change just one specific letter in the RNA, it fundamentally alters how that protein folds. It converts the dangerous APOE4 protein into a benign protective version called APOE1.
Just one letter.
Just one letter. And their new CU5.1 editor achieved this exact change with about 90% efficiency.
Wow. And they also went after ALS, or Lou Gehrig's disease, right? There is a specific mutation in the SOD1 gene that causes a highly toxic protein to build up and destroy motor neurons. The editor found the typo and fixed it at 85% efficiency.
That's staggering.
And for retinitis pigmentosa, which is a disease that causes blindness, they used the editor to do something slightly different. Instead of changing the protein shape, they edited the RNA to introduce a stop codon.
Ah, yes.
This is basically a biological stop sign that tells the cell's machinery to simply halt production and stop building the toxic mutant protein altogether.
And in all of these cases, the AI had perfectly predicted the editing window. Because of how the complex folds, the new ProAPOBEC enzymes consistently make their edit exactly 2 to 5 genetic letters downstream from where the PUF zip code binds to the RNA. It is incredibly predictable.
All right, so let's do another quick bullet recap of this part of the journey. Bullet 1. Native APOBEC is limited to UC targets. Bullet 2. AI, specifically AlphaFold 2, helps scientists play Mr. Potato Head to create custom ProAPOBECs. And bullet 3. We can now target almost any genetic sequence, whether it's GC, CC, or AC.
Perfectly summarized.
Which makes me want to ask you, the listener, Imagine taking the engine of a Ferrari and putting it in an off-road vehicle. That is essentially what AI just did for genetics. How does that change your view of AI in medicine? It's not just a chatbot anymore.
No, it is actively inventing biology. But of course, working in a petri dish is one thing. To prove this could actually be the future of medicine, they had to move into live mammals.
Right.
So they aimed at a major global health issue, high cholesterol, specifically a target called PCSK9.
Okay, so PCSK9 is a protein produced in the liver, and its job is actually a bit counterintuitive. It prevents your body from clearing out the bad cholesterol, LDL-C. So if your liver produces too much PCSK9, your bad cholesterol stays dangerously high. The algorithm the researchers designed to combat this is so elegant, they deliver the editor to the liver. The PUF zip code binds to the PCSK9 mRNA. The APOBEC eraser edits one specific C into a U. And this single edit changes the genetic instruction into one of those premature stop codons we mentioned earlier.
And this triggers a really fascinating biological mechanism called nonsense-mediated decay.
Nonsense-mediated decay.
Right. So the cell's internal machinery is constantly reading its RNA. When it hits an unexpected stop codon right in the middle of a sequence, it realizes the message is corrupted. So it triggers a natural garbage disposal process.
Wait, so it just shreds it?
Yes. The body shreds the mRNA entirely. The PCSK9 protein is never manufactured, and without that protein blocking the way, your body naturally clears out the bad cholesterol.
And here's a detail that I found absolutely delightful. To do this, they screened all of their AI-generated ProAPOBECs to find the absolute most efficient one for this specific liver target. The winner achieved a massive 96% editing efficiency in the lab.
96%.
Yeah.
Yeah.
It was a variant called CU5.21. And where did the core engine for this specific variant come from? Ailuropoda melanolica.
The giant panda.
The giant panda. Through AI simulation, they literally utilized a piece of panda genetics to cure high cholesterol. I mean, you can't make that up.
It's amazing. And to actually get this panda enzyme into the living mice, they utilized a viral delivery system called AAV8.
AAV8?
Yeah, this is an adeno-associated virus. Scientists strip out all the genes that make the virus cause sickness, and what you are left with is essentially a microscopic delivery truck. The reason they chose AAG8 is because it has a natural tropism, or a biological preference, for infecting liver tissue.
Okay, so it homes right in on the liver.
Exactly. They injected this into the mice, and 4 weeks later, the results were staggering. The target mRNA levels in the liver dropped by over 70%, and serum cholesterol plummeted.
And the most vital piece of data regarding safety, they took the genomic DNA from these mice, the permanent hard drive, and deep sequenced it. They scanned the entire genome and found 0 off-target DNA mutations. The hard drive was completely untouched. Only the temporary RAM was edited.
It's a massive success. They proved that by using an AAV virus to deliver an AI-designed editor, they could successfully degrade a disease-causing RNA in a live animal, lowering cholesterol safely.
But I have to ask, the liver is actually one of the easiest organs to target, right? Because it's literally designed to filter the blood. It grabs everything. What about the brain?
The brain is much harder.
Right, because can we use this temporary software update to fix severe neurological conditions? The brain is protected by the blood-brain barrier, which is this incredibly strict biological fortress designed to keep foreign substances out.
It is the ultimate challenge in gene therapy. And the researchers took on a profound clinical case to test this. They looked at a tiny point mutation in the MEF2Z gene. Specifically, it's known as the L35P mutation.
L35P?
Yes. In humans, and in the mouse models they used, this single-letter typo causes severe autism spectrum disorder. And that's characterized by profound social deficits and a significantly shortened lifespan.
So, to get the editor past the fortress of the blood-brain barrier, they packaged their most effective brain-targeting variant, which was CU5.15, into a highly specialized viral delivery truck called AAV-PHP.EB.
Quite a name.
I know, AAV-PHP.EB. But this specific virus was explicitly engineered to cross into the brain, and the crazy thing is, they didn't have to drill into the skull or anything, they just injected it straight into the tail vein of the mice.
And this raises an incredibly important point about the logistics of gene therapy. Previous attempts to cure this exact mutation using permanent DNA editing, like CRISPR, ran into a major physics problem. Way too big. The CRISPR DNA editing machinery is physically huge. It's a massive molecule. It literally cannot fit inside a single AAV delivery truck. You have to split it in half and use 2 separate viral vectors.
Oh, that sounds complicated.
It is. Delivering 2 vectors simultaneously into the brain of a patient and hoping they both infect the exact same cell at the exact same time is incredibly inefficient and poses major clinical risks. But this new RNA editor, the ProAPOBEC, is much more compact. It fits perfectly into a single vector.
That single vector advantage is huge. And the results from that simple tail injection were astonishing. They achieved 30% to 50% CDU editing deep in the brain, specifically in the prefrontal cortex and the hippocampus.
Which is very hard to reach.
Very. And that level of editing was enough to fully restore the normal, healthy levels of the MEF2C protein in those brain regions. But of course, protein levels are just, numbers on a page. The real proof is in the behavior.
Absolutely. So to measure the behavioral impact, they ran the mice through rigorous protocols. One is called the three-chamber test, which measures sociability. It tests whether a mouse prefers to spend time interacting with another live mouse or if it prefers to hide in an empty space. Another is the social intruder test, which measures how they react to novel, unexpected social situations over multiple days. Untreated mice with this MEF2C mutation show extreme social avoidance and anxiety. They isolate themselves completely.
And the treated ones?
The mice treated with the single vector RNA editor completely regain normal social novelty and interaction behaviors. They behave just like the healthy control mice.
And perhaps the most incredible, undeniable finding of the entire study: their lifespans were dramatically extended. A previous study that used permanent DNA editing on this exact same mutation in mice did manage to fix some behaviors, but it failed to extend their lifespan. But this transient RNA editing successfully extended their lives, nearly matching the lifespans of perfectly healthy mice.
It's a monumental proof of concept. By using a specialized virus to cross the blood-brain barrier, this compact single-vector system can effectively treat severe neurological conditions deep in the brain. It restored the vital protein levels, it completely reversed the autism-like behaviors in the mouse model, and it gave them their lives back.
Okay, time for our final recap. Bullet 1. AAV viruses deliver the ProAPOBEC editors to specific organs, like the liver or the brain. Bullet 2. PCSK9 editing successfully lowered cholesterol. Bullet 3. MEF2C editing reversed autism-like phenotypes and extended lifespan in mice.
Spot on.
So think about the implications here, listener. You get an IV injection, and a viral delivery system updates the RNA in your brain to cure a condition. What are the ethical and medical boundaries of this? It's huge.
It really is.
Which brings us to the synthesis of this entire journey. We started today with the simple dream of a genetic text editor, an invisible cursor that could fix typos on the fly. We explored how the rewire system cleverly uses PUF zip codes to read the chemical faces of RNA and APOBEC erasers to make the changes.
Then AlphaFold 2 came in.
Right. AlphaFold 2 AI allowed scientists to play Mr. Potato Head, perfectly combining core engines from humans, mice, and even the giant panda to create ProAPOBECs that break the rigid rules of nature. And finally, we saw undeniable proof that this technology works in living mammals. It can lower high cholesterol in the liver, and it can reverse severe neurological disorders in the deep brain, all without permanently altering the hard drive of their DNA.
And this is exactly why RNA epigenetics and transient base editing represent the absolute frontier of modern medicine. You get the precision of a surgical scalpel with the safety of a temporary bandage. If a treatment goes wrong, or if better technology comes along in 5 years, the patient isn't locked in forever. The edited RNA naturally degrades over time, and you can simply apply a new, updated treatment.
It really is the dawn of the biological software update. So I want to leave you with this final thought to ponder today. If we can temporarily rewrite our genetic messages to boost our health, to clear our cholesterol, or to cure complex diseases without ever altering our permanent genome, will we eventually treat genetic medicine less like a high-stakes permanent surgery and more like an app update we download every few months? And if it truly becomes that easy and that temporary, where do you draw the line between healing a disease and simply upgrading the human machine? Think about that text message we talked about at the start. Soon, you might not just be correcting the typos, you might be rewriting the whole conversation.